So this is where we left off on the last one. There's just one other little piece. Remember, this is the um, pyruvate was created in glycolysis. Now this pyruvate becomes oxidized and then taken into the mitochondria. In the course of breaking down pyruvate into acetyl-CoA, you have CO2 given off, actually a good amount of CO2 given off, and then the electrons, because pyruvate has been electrons have been taken off, they're passed on to the NAD. So then NAD becomes NADH. Because that is a covalent, so it's actually, we're talking about that whole hydrogen ion. And then in step 3 of the oxidation of pyruvate, it's then attached. So you actually have taken carbons off. So the fragment is attached to CoA, so it becomes acetyl-CoA. It actually has only 2 of the carbons. Remember, when it went in, it had 3 carbons. Now it just has 2 carbons. The number of carbons, not so important. When I mention that, I'm just trying to let you know that some bond has been broken, and in the course of that bond being broken, you're going to have most likely an electron released. That electron then needs to be passed off into something. It's going to be passed off into those electron carriers. The citric cycle itself, the citric acid cycle, happens inside the mitochondria. I just brought this particular slide back in. We talked a little bit about the mitochondria for test 1 in chapter 6, but just to refresh your memory, the mitochondria is one of the organelles you find in every cell. Depending on where the cell is in the body, sometimes particularly muscle cells have a tremendous amount of mitochondria, not too many mitochondria in fat cells. So wherever there's the need for a tremendous amount of ATP, you're going to have multiple mitochondria. And the thing about the mitochondria is it's a double membraned organelle. So you have an internal membrane and a space between. So that inner membrane creates a negative space, and that negative space actually is kind of convoluted to make more surface area. We call this the cristae. The cristae is the expanded surface of the inner area of the mitochondrial membrane, and these are where the proteins are attached that then can hold on to those electrons. Henceforth, the cristae in the mitochondria, in the inner membrane of the mitochondria, is where the electron transport chain takes place. The site of actual ATP synthesis is in the matrix. Matrix is location of the reaction of the citric acid cycle, and then so the cristae is the expanded surface of the inner area of the inner mitochondrial membrane, which helps its ability to produce ATP. The matrix is the site of ATP synthesis, also the location of the Krebs cycle or the TCA cycle. In terms of the electron transport in the cristae, most of the components are because of these protein complexes, and this is just a little bit of a bit of information here. This image does not come from your textbook, but I did like this blow up, because what you've done is you've taken what this image does is takes that glucose, it's broken down in glycolysis in the cytosol, moves it into the mitochondria, and this just blows up the mitochondria, where you can see where that pyruvate is then going to be attached to acetyl, and one of the carbons come off, carbon dioxide is going to be released, NAD is going to gain an electron, and then pyruvate is going to be attached to CoA, and it's going to turn twice, because remember you have 2 pyruvates for every glucose in this cycle, and as it turns in this TCA cycle or Krebs cycle, more carbon dioxide is going to be given off, a little bit of ATP is going to be created, but a tremendous amount of electron energy is going to be transferred to the electron transport chain via these electron carriers, which are NAD and FAD. So you can see them in the purple. They're being arrowed down to this area of the mitochondria where you see all these H pluses. So if you can kind of see where the H pluses are, this area where on the outside of the H pluses is where these protein complexes are that then can release the energy. And what happens there is the electron is separated from the proton in terms of hydrogen, and you have way too many hydrogen protons outside the membrane, this inner membrane, and this concept of chemoosmosis is this idea that ATP is going to be created because you have a chemical gradient that is that's going to push those hydrogen ions across the membrane back into the matrix. And we'll look at that a little bit closer, but this particular image shows it pretty well in terms of in an artist's rendition. So this is the pyruvate oxidation image from your book. However, it's the one we're going to use also for the citric acid cycle in terms of the detail that I really need you to know it in. There's an excellent image in your book, and it goes into more detail than we really need you to go into. In terms of from steps 1 to 8, it tells you each step what's given off, and also tells you what the new product that's made is. But the cycle, so that pyruvate is, you're going to remove the carboxyl group, attach it to CoAs, and so we're going to have acetyl-CoA. That acetyl-CoA then goes into the cycle, and you can see it. You can see the acetyl-CoA going into the cycle, and as the cycle turns, one of the things that you do want to put in your head are the products of one turn of the citric acid cycle. One turn is going to create carbon dioxide, 2 molecules of it. One turn is going to create 3 NADs, and those NADs are going to turn into NAD pluses because you actually have that that electron, that hydrogen atom being attached. And also one ADP is going to be phosphorylized into ATP, and then also NAD is going to gain a hydrogen, and that's going to turn twice for every glucose molecule. And so what you have here on your right is the tally per 1 pyruvate, but please understand every glucose molecule is going to create 2 pyruvates. So per 1 pyruvate we have 3 carbon dioxides, 3 NADH, 1 ATP, and 1 FAD. That's in the citric acid cycle. Per 2 pyruvates you have 6 carbon dioxides, 6 NADH, 2 ATP, and 2 FAD. So the thing that you want to have in your head for the test and for the quiz, etc., is the products. And then you have more definition of the products of the glucose molecule. Only 4 of the 38 ATP ultimately produced by respiration of glucose are derived from the substrate level, so substrate level phosphorylation. 2 from glycolysis, 2 from Krebs cycle. This is per glucose molecule. Vast majority, 90%, comes because that NADH and FADH2 are carrying electrons. Those electrons then go into the electron transport chain that then generates the product that causes the phosphorylation of ADP. So this is repeating a little bit of what I just said. It's just I guess repeating exactly what I just said, just to make it a little bit more, to resonate in your head a little bit longer. And then the electrons, electron transport chain, those NAD, then transferred to the first molecule in the chain. And that molecule actually happens to have a name. All of these have names. Not so important that you know that name, but you do want to know that cytochrome proteins are the proteins that are carrying these electrons. So electrons continue along the chain, which includes several cytochrome proteins. We see something similar to this protein in photosynthesis also. These are proteins that actually can carry these electrons. Electrons carried by FADH2 have a lower energy, lower free energy, and are added to the point later. So they're NAD are added first, then FADH2 is added later. They ultimately pass down from protein to protein, and eventually when the energy is low enough, they will be passed down to oxygen and combined with oxygen to form H2O. And that oxygen actually is driving this. It's actually pulling those electrons because it's remember, they're attached to that oxygen. It's the chemical pull of that. It generates no ATP directly. It's important you have that in your head. This chain is not generating ATP. It functions to create large free energy, and free energy that then in the next step is going to be able to, with the help of ATP synthase, this is a enzyme, in the cristae, remember that's the inner membrane, the inner of the inner membrane, the folds, makes ATP from ADP. ATP uses energy from existing protein gradient to power the synthesis. That's why I was saying that to have to pay attention to that image where you had all the H pluses on in the matrix. What's going to happen is there's a need to have equal amounts of H pluses on the outside and inside, so in the mitochondrial matrix in the inner membrane space. So this proton, not protein, but proton gradient is going to move this what we're calling this ATP synthase, is going to cause it to turn. And each turn is going to cause more hydrogens to come in, and in the turning of this complex, this ATP synthase complex, which is being drawn as a rotor here, that energy of the turn, just like a wheel of a wheel in a a mill wheel, which you probably have seen if you've gone on any kind of history field trips, is going to fuel the energy that's going to cause that phosphate to go on to ADP. So each turn is actually going to cause an ATP or get enough energy for the ATP to be created. The molecules are only place that allow hydrogen to diffuse back into the matrix. This flow of hydrogen is used by the enzyme to generate ATP, called chemiosmosis. And chemiosmosis is a term that you do want to have in your head. Chemiosmosis is defined as the oxidative phosphorylation that results in ATP production in the inner membrane of the mitochondria. Now you have a nice summary of cellular respiration. It's a good picture with regard to if you're still kind of not clear about what is produced, and so not clear about what's produced generally, so NAD or FAD or CO2, and also where it's produced. And we'll pick this up on the next one.